Welcome to Biology 1440. This is the first pre-lab video for the first in-person lab of the semester. That lab will be over lab 2 of the manual. And the purpose of this lab, well, there's several purposes, but we're going to learn some experimental design, hypothesis writing, and how to make a standard curve. So cell membranes are found around every known cell. They are comprised of a phospholipid bilayer. So there's phospholipids here and here, making almost like a sandwich. And there are proteins embedded in or stuck to the periphery of the cell membrane. And we are going to look at, specifically this week, beet cells. We're going to look at them this week and in a future lab as well. And we're going to look at beet cells not because we are interested in beets for their own sake, but because beet cells contain a red pigment known as beta-cyanin. And when you damage their cells, that red pigment leaks out, so we can measure it. So if we are interested in asking a question like what kind of substances damage cell membranes, a beet is a good organism to test that on because it has cell membranes that are quite a bit like your membranes or any other organism's membranes. But in the case of a beet cell, you can actually easily tell when they've been damaged because red stuff leaks out. So that's why we use a beet as a model organism. So anytime this semester when we're using a model organism, I might ask you, why is this a good model for the question we're asking and for the experiment we're doing? And you can figure out what makes this particular organism a useful organism to study this particular idea. Now, in the case of a beet cell or any plant cell, they have a cell wall around the outside, and they have a plasma membrane, or that's another word for cell membrane. Don't confuse a cell wall with a cell membrane. A cell wall is composed of lignin and cellulose. It's basically like your desk or your pencil. It's woody. It's a wood. Whereas the cell membrane is more like Crisco. It's a layer of phospholipids. Now, a plant has a cell wall, then a phospholipid layer, and then they have lots of organelles, and each of these organelles has their own phospholipid bilayers. The organelle that we're most interested in for this week is the plasma membrane and then the tonoplast membrane. The tonoplast membrane is the membrane around the central vacuole. So the central vacuole is basically like a big water balloon inside the cell. It holds lots of fluids. It helps keep the cell turgid or rigid when it's full of water. But in the case of a beet cell, it also holds on to that beta-cyanin pigment. So in a future lab, if you were to damage these cells and you were going to measure how much of their cell membranes have been damaged, you might damage the plasma membrane, let's say with alcohol, with ethanol, the stuff that we use to sanitize our hands. We could damage this outer membrane, and then that ethanol could seep in and damage this membrane around the vacuole, and then that red stuff would leak out, and we could measure how much of it leaks out. And we could infer that the more red stuff that leaks out, the more we've damaged those membranes. We've either destroyed the proteins or we've knocked apart the phospholipid bilayer. So our basic goals this week are to learn how to use pipettes to measure and transfer solutions. We're going to use the mass balance equation to make dilutions. We're going to use a spectrophotometer to determine lambda max for a solution. We're going to use the spectrophotometer then to collect beta-cyanin absorbance data using different concentrations of beta-cyanin. We're going to create a standard curve using this data and then use that standard curve to determine the concentrations of unknown beta-cyanin solutions, which is how we could eventually measure how much a cell membrane has been damaged. So beets, once again, have a central vacuole that fills up about 90% of the cell, and that's where that beta-cyanin, that red pigment, is found. The way we can measure how much of that red pigment is present in a solution if we've damaged a cell is using a spectrophotometer. A spectrophotometer uses visible light, which is part of the electromagnetic spectrum. And so visible light ranges from red to violet, or 750 nanometers wavelength to 400 nanometers wavelength. So we measure wavelength, that's the distance from one wave to the next, using wavelength, which is in nanometers, a billionth of a meter. And the symbol of wavelength is lambda, which is looks like an upside down Y. Okay? So red light has the least amount of energy in it. It has 750 nanometers between the waves, whereas violet light has waves that are closer together, around 400 nanometers. So if you have beta cyanin in a test tube or in a cuvette, which is the fancy word for the tube that you put the solution in when you put it into a spectrophotometer. So let's say you have a solution of beta cyanin and you put it in a spectrophotometer. What a spectrophotometer does is it shines different colors of light through that specimen, and it records what passes through and what does not pass through. And what we're actually interested in is not what color is reflected by beta cyanin, that red pigment, but we're interested in what color is absorbed by beta cyanin and which wavelength is most absorbed by that solution. And so the wavelength that is most absorbed by a substance is known as the lambda max, the wavelength most absorbed by that solution. And that's what we will use to determine the concentration of beta cyanin in a solution. It's really strange. You can use how much light a solution absorbs. You can use that data to calculate the actual concentration of the of the solution. Okay. So I'll explain how that works briefly in a moment, and you'll learn more about it in lab. But before we do that, I just want to mention that we're going to be testing hypotheses in Bio 1440, and I typically ask that you write your hypotheses in a research hypothesis format, which is if and then. If is your explanation. Technically, it's the part that really is the hypothesis. A hypothesis is an explanation. After the word and, you tell the reader what your basic experimental setup was, followed by the word then, which is what you predict will happen if your explanation is right. So for example, for this week, we might say, if light absorption by a solute increases directly as the solute concentration increases, and the absorbance of a variety of beta cyanin concentrations are determined using a spectrophotometer and graphed, then it will be possible to determine the solute concentration in any solution by measuring the amount of light absorbance in that solution. I have a typo there. In that solution. If and then. Now, if we want to tone this down, you can say, if the, let's just tone this way down for a second. I would expect you to write more like this paragraph, but let's tone it down for a minute. If the amount of light that beta cyanin absorbs changes if it's in a higher or lower concentration, and you test a variety of beta cyanin solutions, then basically solutions with more beta cyanin will be darker and absorb more light, and solutions with less beta cyanin will absorb less light, right? And therefore we can quantify that and determine how much light is being absorbed, and then we can use that to determine how much beta cyanin there is in the solution. Now, how in the world does that work? Well, that works with the use of a standard curve, which you're going to make using your data from lab this week. A standard curve is a graph. Basically what you do is you have a known concentration of, in this case, beta cyanin. You're going to make different concentrations of beta cyanin. This might go wrong. PowerPoint likes to put weird lines when I draw. But here's an XY axis. So you're going to test known concentrations of beta cyanin. That's what's down here. That's going to be our independent variable. And then light absorbance, I'm just going to put an A here. That's absorbance. That's how much light is absorbed by these things. So you'll have known concentrations. You'll have different concentrations here, and you're going to see how much light they absorb, right? So these are knowns or standards, and you're going to have a graph that you put together with this. Oh, that's terrible. Anyway, and once you do that, if I give you an unknown concentration of beta cyanin, maybe it falls right here, you can determine what concentration that unknown solution is using your standard curve. We can do that in two ways, through interpolation or extrapolation. So if I give you, let's say your standards were between these two red lines, right? If I give you an unknown that falls right here, that falls within the range of your original standard curve. So determining its concentration is known as interpolation. If I give you an unknown concentration that's way less or way more than the standards you use to make that curve, that's extrapolation. It'll be a little less accurate, but you can still make a prediction. You can make a prediction as to what you think the concentration of that unknown is using the standard curve. So you might build a standard curve that looks like this. You might write a caption underneath it. The other lab you'll be doing online, either before this or after this, depending if you're in group A or B, you'll learn how to make a figure like this. And here's the standard curve. Here's the equation of the line. You can solve for the unknowns using the equation of the line, or you can do it by drawing a line onto your graph. For example, let's say I gave you an unknown concentration and it had an absorbance of 0.7. Once again, the lines might get weird here, but I could draw a line over, using a straight edge of course, over to where this intersects the graph and down. And where it intersects the x-axis, I could say, oh, the unknown concentration is maybe 13 micromolar, let's say. Okay, so we can use the standard curve to determine what an unknown concentration of beta-cyanin was. So this week in lab, you're going to learn how to do all of this. So read the lab, paying special attention to the mass balance equation, how to do that. There's going to be a pre-lab quiz that you're going to take probably online. So you'll want to read this ahead of time so you're ready for that pre-lab quiz. You'll likely take that quiz before you even get to lab, depending on how your lab instructor decides to do it. All right, good luck everybody. Have a good week.